Watson and Crick, discovering the structure of life. A deep sleep journey into one of science's greatest discoveries. Welcome to the Deep Sleep Channel. Tonight we're exploring one of the most profound discoveries in the history of science, a discovery that revealed the fundamental code of life itself. A discovery that answered one of humanity's oldest questions: how does life work? How does heredity pass from parent to child? What is the physical basis of inheritance? This is the story of the DNA double helix. In 1953, two young scientists named James Watson and Francis Crick made a discovery that would revolutionize biology and change our understanding of life itself. They discovered the structure of DNA, the molecule that carries the genetic code of all living things. The discovery was not the result of a single brilliant experiment. It was the result of years of work by many scientists. It was the result of X-ray crystallography by Rosalind Franklin. It was the result of chemical analysis by other researchers. It was the result of brilliant theoretical thinking by Watson and Crick. But when Watson and Crick finally figured out the structure, when they finally understood how the atoms were arranged, they realized that they had discovered something extraordinary. The structure was elegant, the structure was simple, the structure was beautiful, and the structure immediately suggested how genetic information was stored and how it was transmitted from one generation to the next. Close your eyes. Let yourself travel back to 1953. Let yourself meet these brilliant scientists. Let yourself witness the moment when the structure of DNA was revealed. Let yourself understand the beauty and elegance of this molecule. Let yourself contemplate what this discovery means for life itself. Let yourself drift into sleep, understanding that within every cell of your body, this elegant structure is working, passing down information from generation to generation, making you who you are. This is a meditation on discovery, on the power of human intellect, on the beauty of science, and on the fundamental nature of life itself. The question: what is life made of? To understand the significance of the DNA discovery, we must first understand the questions that scientists were asking in the early 20th century. Life is extraordinary. Living things grow, they reproduce, they pass on characteristics from parent to offspring. A child inherits traits from both parents. A puppy grows up to look like its parents. A plant grown from a seed develops into a plant that looks like its parent plant. How does this inheritance work? What physical mechanism allows traits to pass from parent to child? This question had fascinated scientists for centuries. In the 19th century, Gregor Mendel discovered the laws of heredity through his careful experiments with pea plants. Mendel showed that traits are inherited in predictable patterns. Some traits are dominant, some are recessive. Traits follow mathematical ratios when plants are bred together. But Mendel's work raised new questions. What was the physical basis of these inherited traits? If traits followed mathematical patterns, there must be some physical mechanism that caused these patterns. But what was it? Scientists knew that the hereditary material was somehow located in cells. They knew it had something to do with chromosomes, the structures inside the nucleus of cells. But what exactly was the hereditary material made of? How was information stored in it? How was that information used to build living organisms? In the early 20th century, scientists began to identify the chemical components of cells. They discovered that cells contained proteins, which were made up of amino acids linked in chains. They discovered that cells contained other molecules, including a substance called nucleic acid. The nucleic acid was particularly interesting. Scientists found that nucleic acid was concentrated in the nucleus of cells, the place where chromosomes were located. This suggested that nucleic acid might be the hereditary material. But how could nucleic acid carry the complex information needed to build a living organism? Nucleic acid seemed to be a simple molecule, made up of only a few components. How could something so simple carry such complex information? The chemistry of DNA, understanding the building blocks. To understand DNA, we must first understand the chemistry of life. Cells are made up of molecules. Molecules are made up of atoms. The atoms in living things are primarily carbon, hydrogen, oxygen and nitrogen, with smaller amounts of phosphorus, sulfur and other elements. These atoms link together to form molecules. The molecules form larger and more complex structures, and those structures form the living cell. One of the most important types of molecule in living things is the protein. Proteins are made up of amino acids linked together in long chains. There are 20 different amino acids that can be linked together. Different combinations of amino acids, in different orders, create different proteins with different functions. Some proteins are enzymes, which speed up chemical reactions. Some proteins provide structure to cells. Some proteins transport other molecules. Some proteins regulate other biological processes. The diversity of proteins comes from the different orders in which amino acids can be linked together. Another important molecule is nucleic acid. DNA is a type of nucleic acid. DNA is made up of smaller units called nucleotides. Each nucleotide consists of 3 components: a sugar called deoxyribose, a phosphate group, and a nitrogenous base. There are 4 different nitrogenous bases in DNA. They are adenine, thymine, guanine, and cytosine. Often they are abbreviated as A, T, G, and C. These 4 bases are the letters of the genetic alphabet. They are the symbols used to write the genetic code. The nucleotides link together to form long chains. The sugar and phosphate groups form the backbone of the chain. The nitrogenous bases stick out from the backbone. Different sequences of bases create different messages, just as different sequences of letters create different words. But the chemistry of DNA was not fully understood in the early 20th century. Scientists knew that DNA was made up of nucleotides, but they didn't know how the nucleotides were arranged. They didn't know how the DNA molecule was structured. Without understanding the structure, it was impossible to understand how genetic information was stored or how it was used. Rosalind Franklin, the X-ray crystallographer. To discover the structure of DNA, scientists needed a way to see inside the DNA molecule. They needed to know how the atoms were arranged in 3-dimensional space, but DNA molecules are much too small to see with any ordinary microscope. The solution was X-ray crystallography. X-rays are a type of electromagnetic radiation with very short wavelengths. Wavelengths so short that they can pass between atoms in a crystal. When X-rays pass through a crystal, they are scattered by the atoms. The scattering pattern can be captured on photographic film. By analyzing the pattern, scientists can figure out how the atoms are arranged. A scientist named Rosalind Franklin became an expert in X-ray crystallography. Franklin was born in 1920 in London. She was interested in science from an early age. She studied physics and chemistry at university. After graduating, she worked as a researcher, studying the structure of various molecules using X-ray crystallography. 1951, Franklin joined a research group at King's College London. Her task was to use X-ray crystallography to determine the structure of DNA. Watson and Crick, the brilliant theoreticians. While Rosalind Franklin was doing careful experimental work, 2 other scientists were thinking about the structure of DNA from a theoretical perspective. James Watson was an American biologist born in 1928. He was a precocious student. He entered the University of Chicago at the age of 15. He was interested in genetics and wanted to understand the physical basis of heredity. After earning his doctorate, he spent time in Europe, learning about DNA research and meeting leading scientists in the field. Francis Crick was a British physicist born in 1916. Crick had initially trained in physics, but after World War II he became interested in biology. He was fascinated by the question of how genetic information was stored in molecules. He believed that if you could understand the structure of the genetic material, you could understand how life worked. Watson and Crick met in Cambridge, England in 1951. They began working together, thinking about the structure of DNA. They were not doing experiments themselves. Instead, they were thinking about the problem, using available data from other scientists and trying to figure out what structure would make sense. They knew that DNA was made up of nucleotides. They knew that the nucleotides were linked together by sugar and phosphate groups. They knew that the DNA molecule had some kind of helical structure based on Franklin's X-ray photographs. But they didn't know exactly how the pieces fit together. Watson and Crick made models. They built physical models using wire and cardboard, trying to figure out how the atoms could be arranged. They thought about which arrangements would be chemically stable. They thought about which arrangements would allow genetic information to be stored and retrieved. They were aware of Chargaff's rules, which stated that in DNA, the amount of adenine equaled the amount of thymine, and the amount of guanine equaled the amount of cytosine. This suggested some kind of pairing between the bases. But how were they paired, and what did this pairing mean? The crucial insight: base pairing. The breakthrough came when Watson and Crick realized how the bases must be paired with each other. Watson realized that adenine and thymine had similar shapes, as did guanine and cytosine. If the bases were paired in this way, with adenine pairing with thymine and guanine pairing with cytosine, then the width of the DNA molecule would be uniform. The pairs would all be the same width, which would allow the bases to fit snugly together. This base pairing also explained Chargaff's rules. If adenine always paired with thymine, then the amount of adenine would always equal the amount of thymine. If guanine always paired with cytosine, then the amount of guanine would always equal the amount of cytosine. The chemical rules and the base pairing explained each other. But the base pairing did more than just explain the chemical rules. It suggested something profound about how genetic information was stored and how it was copied. If one strand of DNA had the sequence ATGC, then the paired strand would have to have the sequence TACG. The bases on one strand would determine the bases on the other strand. This meant that if you knew the sequence of bases on one strand, you could predict the sequence of bases on the other strand. And this had an obvious implication for DNA replication. If the two strands could be separated, each strand could serve as a template for making a new complementary strand. One DNA molecule could be copied into two DNA molecules, each containing one original strand and one newly made strand. Watson and Crick immediately saw the significance of this. They understood that the base pairing mechanism provided a way for genetic information to be accurately copied and passed from one generation to the next. The structure, the double helix. Now Watson and Crick began to put all the pieces together. They thought about Rosalind Franklin's X-ray photographs which showed a helical structure. They thought about the base pairing. They thought about the sugar-phosphate backbone linking the nucleotides together. They visualized two strands of nucleotides wound around each other in a helix. The sugar-phosphate backbone was on the outside of the helix. The nitrogenous bases were on the inside, paired with each other. Adenine paired with thymine, guanine paired with cytosine. The structure was elegant, it was simple, it was beautiful, and it immediately suggested how genetic information was stored. The sequence of bases could be read as a code. Different sequences meant different information, and the base pairing mechanism meant that the information could be accurately copied. Watson and Crick built a detailed model of the DNA structure. They calculated the angles and distances between atoms. They checked that all the bond angles and distances were chemically reasonable. They made sure that the model was consistent with all the available experimental data, including Franklin's X-ray photographs. The model showed two strands wound around each other in a right-handed helix. The strands were anti-parallel, meaning that they ran in opposite directions. The bases were on the inside, stacked on top of each other, paired with bases on the other strand. The structure was stable and elegant. The announcement. April 25, 1953. On April 25, 1953, Watson and Crick published their findings in the journal Nature. They presented the structure of DNA. They explained the base pairing. They suggested how the structure would allow for DNA replication. The paper was relatively brief, only about 1,000 words, but it was one of the most important scientific papers ever published. It launched a revolution in biology. At the end of the paper, Watson and Crick wrote a now famous understated sentence: "It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material." This sentence was a masterpiece of understatement. They were describing one of the most important insights in the history of science. They were explaining how life could reproduce itself. They were answering one of humanity's most fundamental questions, but they wrote it as if it were a minor observation. The discovery was met with enormous excitement in the scientific community. The structure made sense. It explained the chemical rules that scientists had observed. It suggested a mechanism for DNA replication. It provided a physical basis for understanding heredity. Understanding the implications. How DNA works. Once the structure of DNA was understood, scientists could begin to understand how it functioned. How did the information in DNA lead to the development of a living organism? How did the genetic code work? Scientists realized that the sequence of bases in DNA could be read like a code. Different sequences of bases specified different proteins. The code was written in 3-letter words. Each 3-letter word, called a codon, specified a particular amino acid. By reading the codons in sequence, a ribosome could assemble amino acids in the correct order to build a protein. The process was more complex than just reading the DNA sequence. The DNA sequence was first transcribed into a related molecule called RNA. The RNA was then translated into a protein, but the fundamental principle was the same. The sequence of bases in DNA determined the sequence of amino acids in proteins, and the proteins determined the characteristics of the organism. This understanding of the genetic code was revolutionary. It showed that all living things used the same basic code. A codon that specified a particular amino acid in a human specified the same amino acid in a bacterium. This universality of the genetic code suggested that all life on Earth was descended from a common ancestor. The structure of DNA also suggested how mutations could occur. If an error was made during DNA replication, one base might be substituted for another. This would change the sequence of bases in the DNA. This could lead to a change in the protein that was produced. This could change the characteristics of the organism. This was the physical basis of mutation and evolution. The replication story. How life copies itself. One of the most beautiful aspects of the DNA structure is how it suggests a mechanism for replication. Watson and Crick immediately understood this, and subsequent experiments confirmed their prediction. The double helix can be unzipped, the 2 strands can be separated from each other. When this happens, each strand is exposed. Each strand has bases sticking out from the sugar phosphate backbone. These bases are unpaired, but the bases don't stay unpaired for long. New nucleotides come along and pair with the bases on the separated strands. Adenine pairs with thymine, thymine pairs with adenine, guanine pairs with cytosine, cytosine pairs with guanine. As the new nucleotides pair with the bases on the separated strands, they are linked together by enzymes to form new sugar phosphate backbones. 2 new strands are synthesized. Each new strand is complementary to the old strand it is paired with. The result is 2 DNA molecules, each identical to the original. Each new DNA molecule consists of 1 original strand and 1 newly synthesized strand. The genetic information has been perfectly copied. This process is called semi-conservative replication because each new DNA molecule conserves 1 of the original strands. The mechanism is so elegant and so efficient that it allows DNA to be copied with almost perfect accuracy, millions of times in every organism. The replication process is the key to how life reproduces. When a cell divides, its DNA is replicated. Each daughter cell gets an exact copy of the genetic information. The information is passed on faithfully from one generation to the next. The scientific method in action. How the discovery came about. The discovery of the DNA structure is a beautiful example of the scientific method in action. It shows how different scientists contribute different pieces to the puzzle. It shows how theoretical thinking and experimental work complement each other. It shows how brilliant minds can work together to solve difficult problems. Rosalind Franklin provided crucial experimental data through her X-ray crystallography. She was meticulous, she was careful. She collected high-quality data. Without her data, the structure could never have been determined. Watson and Crick provided theoretical thinking and intuition. They could see connections that others might have missed. They could imagine structures and test them mentally. They could integrate different pieces of information into a coherent whole. Other scientists like Linus Pauling contributed knowledge about chemistry and how atoms bond together. They provided principles that Watson and Crick could use to evaluate whether their proposed structures were chemically reasonable. The discovery was not the work of a single person. It was a collaborative effort. It was a triumph of human intelligence working together. The reception. Initial skepticism and growing acceptance. When Watson and Crick first published their proposal, not everyone accepted it immediately. Some scientists were skeptical. The evidence was strong, but it was not absolutely conclusive. Some scientists wanted more experimental confirmation. But as time went on, more and more evidence accumulated. Other scientists confirmed that the structure made sense. Experiments showed that the base pairing was correct. The mechanism of semi-conservative replication was experimentally demonstrated. The genetic code was deciphered, showing exactly how the sequence of bases specified the sequence of amino acids. By the late 1950s and early 1960s, the DNA structure was widely accepted. It became the foundation for modern molecular biology. And now does Lu Nandasa Rumbi Adanamstan Rumbi Anara Nandasa Rumbi Nansa Amano Nanasa Nandasa said the Sawaza Trasa. The Thames scientists have remarked that the DNA structure is not just scientifically correct, but aesthetically beautiful. The structure has a rightness to it, a feeling that this is the way it must be. This connection between truth and beauty is something that many scientists experience. They find that the most correct theories are often also the most elegant and beautiful. There seems to be a deep connection between the way the universe actually works and the aesthetic sense of what seems beautiful and harmonious. The human story. Watson, Crick, and Franklin. Behind the discovery of the DNA structure is a human story. It is a story of brilliant minds. It is a story of ambition and competition. It is a story of scientific collaboration. It is also a story with some shadows. James Watson and Francis Crick were highly ambitious. They wanted to make a major discovery. They wanted to be remembered. This ambition drove them to work hard on the DNA problem. It drove them to think deeply about the available evidence. It drove them to propose a bold and beautiful structure. Watson and Crick were also competitive. Other teams of scientists were working on the DNA structure. Linus Pauling in the United States was a leading candidate to solve the problem. Watson and Crick knew they were in a race. They wanted to reach the solution first. Rosalind Franklin was a careful, methodical scientist. She was not interested in rushing to conclusions. She was interested in collecting good data. She was interested in understanding the detailed structure of DNA. She was not the type to propose bold structures based on limited evidence. There is some historical debate about whether Watson and Crick used Franklin's data appropriately. Franklin's work was presented to them, and they used it in their thinking. But it is unclear whether they fully understood at the time how crucial her work was to their breakthrough. After the discovery became famous, Watson and Crick received most of the credit and recognition. Franklin, who had done crucial experimental work, received less recognition. Some historians believe this was unfair. But ultimately, the contribution of each scientist is undeniable. Franklin provided the crucial experimental data. Watson and Crick provided the brilliant insight into what structure would explain that data. Together, and with contributions from many other scientists, they solved one of science's greatest mysteries. DNA today. From discovery to the genome. Since Watson and Crick's discovery in 1953, our understanding of DNA has deepened enormously. Scientists have learned how to read DNA sequences. They have learned how to manipulate DNA. They have learned how to use DNA information to understand life. One of the greatest achievements in modern science is the sequencing of the human genome. The human genome is the complete set of genetic information in a human being. It consists of about 3 billion base pairs. It contains about 20,000 genes. Each gene is a sequence of DNA that codes for a particular protein or RNA. Scientists from around the world collaborated on the Human Genome Project, which took over a decade to complete. In 2003, they announced that they had sequenced the entire human genome. They could read the complete genetic code for a human being. This achievement opened up entirely new possibilities for understanding human biology and disease. Scientists could now identify which genes were associated with particular diseases. They could understand how genetic variations between individuals led to differences in appearance, metabolism, and disease susceptibility. The sequencing of the human genome also revealed something unexpected. Humans have far fewer genes than scientists had expected, but the genes are more complex. They can be regulated in many different ways. They can be spliced together in different configurations to produce different proteins. The relationship between genes and proteins is not as simple as initially thought. The study of DNA has also led to an understanding of evolution at the molecular level. By comparing the DNA sequences of different species, scientists can determine how closely related different organisms are. They can trace the evolutionary relationships between organisms. They can see how genes have been conserved across millions of years of evolution. The power of understanding, knowledge, and responsibility. The discovery of the DNA structure and the subsequent explosion of knowledge about genetics and molecular biology has given humans unprecedented power. We can now understand life at the molecular level. We can read the genetic code. We can manipulate genes. This power brings responsibility. We can use genetic knowledge to develop new medicines. We can develop crops with improved traits. We can understand and potentially cure genetic diseases. But we can also misuse this knowledge. We can create organisms that are dangerous. We can use genetic information in ways that are unethical. The discovery of DNA structure has led to important ethical questions. Should we edit human genes? Should we create designer babies? Should we genetically modify organisms for food? Should we patent genes? These are complex questions without easy answers. But perhaps the most important gift that the discovery of DNA has given us is understanding. We now understand at a fundamental level how life works. We understand how heredity works. We understand how information is stored and transmitted from one generation to the next. We understand our connection to all other life on Earth because we all share the same basic genetic code. This understanding can inspire us to be better stewards of life. It can help us appreciate the complexity and elegance of living things. It can help us understand that we are part of a vast web of life, all connected through the DNA that we share. A meditation on discovery. The joy of understanding. As we drift into sleep, contemplating the discovery of the DNA structure, we contemplate the joy of understanding. There is something deeply satisfying about solving a puzzle. There is something joyful about understanding something that has been mysterious. When Watson and Crick finally understood the structure of DNA, they experienced this joy. They had solved a great mystery. They had understood the physical basis of life itself. They had discovered something so elegant and so beautiful that it immediately felt right. The discovery of DNA structure reminds us that the universe is more orderly and more beautiful than we might imagine. It reminds us that underneath the apparent complexity of life, there are elegant principles. It reminds us that human intelligence can understand even the deepest mysteries of existence. The discovery also reminds us that knowledge is precious. The knowledge of how DNA works has led to advances in medicine, agriculture, and technology that have improved human life. It has led to an understanding of ourselves and our place in the natural world. The continuing story. DNA and the future. The story of DNA does not end with Watson and Crick's discovery. It is an ongoing story. New discoveries about DNA are being made all the time. New technologies are being developed to read and manipulate DNA. New applications of genetic knowledge are being discovered. Scientists are now working on using genetic engineering to cure diseases. They are developing gene therapies that can correct genetic defects. They are learning how to prevent genetic diseases before they develop. Scientists are also working on understanding epigenetics, how genes are regulated and expressed. They are learning that the sequence of DNA is not the only thing that matters. The way genes are turned on and off is also important. This understanding is leading to new insights into how organisms develop and how diseases occur. The field of synthetic biology is developing technologies to create new organisms with design functions. Scientists are learning how to combine DNA from different organisms in new ways. They are creating organisms that can produce medicines, break down pollutants, or perform other useful functions. These developments raise important questions. How should we use this technology? What are the ethical limits? What are the risks? But they also offer tremendous potential for improving human health and solving pressing problems. The structure and the spirit. Why this discovery matters. Why does the discovery of DNA structure matter so much? Partly it matters because of what it tells us about how life works. Partly it matters because of the practical applications that have followed from this discovery. But perhaps most fundamentally, it matters because of what it tells us about ourselves. The discovery of DNA structure tells us that we are made of molecules, that our bodies, our minds, our very selves are the result of physical and chemical processes that occur in your brain, and that despite this, you are capable of understanding the deepest mysteries of existence. Sleep knowing that the DNA structure is a symbol of human achievement. It represents the power of human intelligence to understand life itself. It represents the beauty and elegance of nature. It represents the endless possibilities for knowledge and discovery. Sleep peacefully, contemplating the mystery and the beauty of life. Sleep knowing that the universe is more wonderful than we can imagine. Sleep knowing that within us, in our cells, is a code that links us to all other life on Earth. Sleep knowing that the story of DNA continues. New discoveries are being made, new mysteries are being solved. The journey of understanding is ongoing. Good night from the Deep Sleep Channel. Sleep well and appreciate the beauty and elegance of the DNA double helix. Sleep well and marvel at the mystery of life. Sleep well and dream of the discoveries yet to come.